right. I think everybody's loading in, and we're going to crossfade from the little intro video. So welcome everyone to the webinar, Unearth the Essential Endophytic Fungi. This is part of a larger series that we're putting together, and I'm really excited to be here today and learn from Sam and Kyle. So I just want to let everybody who's joining us today know the pretty much the guidelines that we're following with this Zoom call. So except for the panel, everybody will be muted for the duration of today's webinar. And you have on your Zoom panel options for the chat as well as a Q&A section. If you would like in the second half of our webinar to have one of your questions discussed by the panel, please put it in the Q&A section. The chat is there basically for you to converse with each other as participants. And sometimes, one of us will monitor the chat as well, and we might interact there. And in fact, as per our tradition, I'd love for, some people are already doing it, I'd love for everybody to introduce where they're zooming in from today. So I see Canada and all over Europe coming up already, Uganda, the Czech Republic, various states, including Idaho, where I'm zooming in from. So we hope you have a good time with us today. This is just to give you a little bit of a heads up. This is our introduction phase, and then we will be hearing from Sam for about 30 minutes and from Kyle for about 15 before we go to our Q&A with the panelists. And before we get too much further in here, I think that we would love to hear an introduction from Dr. Elaine, and then we'll just go in this order. So after Elaine finishes, Sam, then you and Kyle, and I can just briefly introduce ourselves. I think you might be muted, Elaine.
I was unmuted. Got it done though. Okay, so I am Dr. Elaine Ingham. I grew up in Minnesota, so the land of 1,000 lakes. It's really more like 20,000 lakes, but close enough. I did my undergraduate degree at St. Olaf College in Minnesota, Texas A&M for my master's degree, where I looked at the digestive system of oysters and figured out the species of bacteria that's required for mussels to be able to get food from what they're ingesting. So kind of fun learning where it would grow and where it would not, and somehow or another in places that have been treated with really toxic chemicals, you don't find them anymore. Don't find the organisms that allow the mussels to grow. So from there to Colorado State University, where I started working with soil. Shock when you go from aquatic systems to soil, because you've got all of this nasty sand, silt, and clay to deal with, rocks and pebbles and all of that stuff. So I've been working on the community of organisms in soil as compared to dirt. Very important difference. So hopefully we'll get around to talking about that. I was the person who worked with Bill Hunt to organize the soil food web pictures and put some data to the information so that we can quantify what we're working with. And I've been working there trying to understand fungal to bacterial biomass ratios. So lots of work yet to do. Nobody can step away. Okay, next.
Hi, everyone. My name is Sam David. I studied biology both in my undergrad and grad school at the University of Wisconsin-La Crosse, which is where I met Kyle. We were in the same grad program. I'll talk a lot more about my thesis research involving endophytes and sedges. But outside of that, I I like to garden, love the outdoors. Yeah, I self-described naturalist and trying to understand whole ecological systems and understand that us as humans look at stuff as good and bad, but when you look at an entire system, good and bad is sometimes irrelevant depending on which way you look at it. Yeah, so excited to be here. Thanks.
Okay, my name is Kyle Kaczynski. For my academic background, I did my undergraduate in plant biology with a minor in chemistry because I was very interested in medicinal plants and how the physiology of plants, how they grew and what made them grow. From there, I became very interested in mushrooms and decided to pursue a graduate program in Wisconsin, which is where I met Sam. We had the same advisor, Dr. Tom Volk, RIP. We really connected in his lab as lab mates. You know, we were working close together, even though our thesis projects were substantially different, I would say. They were both obviously related to fungi, and we were both studying mycology. My particular thesis was more on mushrooms and mushroom cultivation, and then after finishing my master's program, I moved out to Oregon. I'm up here in the Pacific Northwest, where I have a consulting company. We work a lot with farmers growing plants and also teaching them how they can create more sustainable and regenerative systems by incorporating fungi into the soil and into other parts of their system, whether that be mushroom producing fungi or more so soil fungi or endophytic fungi like we'll be talking about today. I say this frequently, but I think it's important to note, especially here at the beginning of the talk, is that all mushrooms are fungi, but not all fungi produce mushrooms. So oftentimes in mycology, mushrooms get the real center stage. And to be honest, that's what brought me to mycology. But the field of mycology is much more vast than just mushrooms. And a lot of these soil fungi and endophytic fungi we don't often see. So our goal for this webinar was just to bring a little bit more awareness to these fungi that are everywhere and that can influence plant growth and ecosystem growth, really. To make sure that more people are aware of this unseen world. So we will definitely be discussing this in more detail as the webinar goes on, but just kind of wanted to give that as a brief intro.
Excellent. Thank you, Kyle. And Sam and Elaine. I'm so happy to be here in the room with such wonderful nerds. And I'm excited about learning from all of you today. So I'm Dr. Adam Cobb. You may have, if you're joining us for, this webinar is not your first, you may have seen me as a panelist, presenter, and host in previous webinars. I do content creation and science communication here at the school. Just recently hit my 2-year anniversary mark working here at the school, and it's been lovely. I had a career before this for a little over a decade as a researcher of arbuscular mycorrhizal fungi, especially in agri-ecosystems. So what Kyle has said is music to my ears because my favorite fungi don't produce mushrooms. And a lot of times people assume that they do. So I'm excited to get into that. And I think from here, Sam, we're going to let you take control as our first presenter and let it rip.
All right. Okay, so yeah, like I said, this is mostly about the research I did as part of my thesis, where I was basically hunting for dark septate endophytes in a particular plant and a particular habitat. But before we get into that, I'm going to do a little background information so we're all kind of at the same starting place. To begin with, I should start with acknowledgements. Thank you to Dr. Lane's Soil Food Web School and Adam and Sammy and Dr. Lane, everybody who's helped put this together. Thank you for Kyle for inviting me to join you on this. It was his idea to share this, so it's been a while for me. It's good to talk about again. I'd like to thank my advisors and help, Tom Volk, Todd Osmondson, Meredith Thompson, Beth Paluch, and Paul Ryerson. They're all really important parts of me doing my thesis research. I'd also like to thank UWL and the RSEL grant for funding my proposal, and for Fort McCoy for allowing me to do my studying on their military base. So to start off with, I'm going to do a little fungal taxonomy. So fungi are defined or split into phyla based off of the sexual spore that they produce. Chytridiomycota produce a swimming spore. Zygomycota produce this hard, large, thick-walled zygospore. Glomeromycota make a big glomerospore. Ascomycota, they produce their spores in a sac-like structure called an ascus. And Basidiomycota produce their spores on a club-like structure called a basidia. And some of the fungi that we're going to be talking about, most people know them as molds or imperfect fungi. And if all mushrooms, or all fungi, I should say, are defined based off of their sexual spore, imperfect fungi don't fit because they don't produce sexual spores. With morphological techniques, we are now able to assign phylogeny based off of genetic testing. Turns out a lot of the molds are Ascomycota, but yeah, that's getting more technical. Okay, so remember these phyla as we talk about different groups as we come. So mycorrhizae, which I'm sure most of you are familiar with, are fungi that develop mutualistic relationships with plants. Typically, they give nutrients such as phosphorus and nitrogen to the plant in exchange for sugars. And that's kind of the basic rundown. And mycorrhizae, again, talking kind of more generalities, are mostly split into ectomycorrhizae, which grow on the outsides of the roots of plants and grow what's called a hard egg net or a mantle. And they are typically Basidiomycota and Ascomycota, which are considered more higher levels of fungi because they have more advanced structures. Endomycorrhizae are also called arbuscular mycorrhizal fungi because they form tree-like structures inside of the cells that look like, well, arbuscule comes from like little tree. So it's these little tree-like structures that grow inside of root cells. And these are almost exclusively glomeromycota. It'll be good to talk with Adam about it later because that's his area of research. But to the best of my knowledge, all AMF are glomeromycota. This is a little bit different than the endophytes that we're going to be talking about. And I'd also like to jump in here and say that endophyte literally means inside plant. And in this particular talk, we're going to be talking about endophytic fungi, but there are also endophytic bacteria, and basically anything that grows within a plant could be called endophytic. But for ease, we're just going to call everything endophytes. They grow in tissues of plants and do not cause harm. Sorry, having a little, I gotta adjust a couple things. Sorry, guys. All right, so endophytes grow inside of tissues of plants without causing harm. They are Ascomycota mostly, and they will grow in what are considered non-mycorrhizal plants. And so there are a few groups of plants that are typically considered non-mycorrhizal. These include brassicas, or a lot of mustards, sedges, and chenopods. And there's been more research showing that some of them may be more mycorrhizal than we thought, including my data. So a little bit more on endophytes. Like I said, they are fungi that grow inside plants without causing harm. In the picture is a fungus called Claviceps purpurea or ergot. And it is a good example of an endophyte because although it does consume the ovary of one of the grains, it provides kind of a chemical defense to the plant. So there are alkaloid compounds inside the fungus. So if an animal or an herbivore were to be grazing and eat this fungus, it's going to get sick and not eat that plant again in the future, ideally. And ergot is an example of how we can get medicine from endophytes. Big one from ergot is medication used to help stop bleeding after childbirth. Another theme that we're going to be talking about in this discussion is endophytes span the continuum from parasite to mutualist. So a lot of times when we think about symbioses in our heads, it's really cut or dry. It's either beneficial or harmful. But these relationships are always in flux depending on environmental conditions and the species involved. So the actual cost benefit of plant fungal interactions is constantly in flux. So in a perfect soil conditions or in a hydroponic setup, a lot of plants that require mycorrhizal fungi don't need fungi. And so the plants basically are contracting out these fungi that are better at acquiring nutrients than they are, so it's more cost effective for them. But these fungi, even if they are not doing direct nutrient exchange, can do other benefits to the plant to help them survive extreme conditions such as fire, pollution, salts, heat, different things of that nature. Again, just illustrating some of the different things endophytes can help protect plants against: heat, salt, oils, and other types of environmental contamination. Endophytes have potential for helping in habitat restoration. Some studies have been performed and shown that endophytic fungi can help with germination and help with other stress tolerances. The applications of these endophytes can also be applied to agriculture. There was a researcher in Tom Volk's lab before us, so same lab as we were in, who looked at endophytes in some crop plants, tomato, barley, and eggplant. They didn't come up with a lot of conclusions as it was a master's project, not a PhD, but it did provide some insight into potential uses. Endophytes can protect plants against pathogens. So bananas and cocoa plants are two examples of crops where there are existing biocontrol endophytic fungi to help them protect them against different pathogens. So getting more into endophyte classification, they have been classified by a research group in a few different ways based on what they grow in, how they grow, and plant host. So is it growing in stem or root? Is it systemic or growing through the whole plant? Or is it localized to, say, just the roots? And yeah, is it in a grass or is it not in a grass? Because some endophytic fungi are specific just to grass, just like Claviceps purpurea or ergot. So I'm going to go through these kind of fast, just because we got a lot to get through. Claviceptaceous endophytes are found throughout grass stems and leaves and into the roots. They are vertically transferred sometimes and provide chemical defenses to the host, like ergot, like we mentioned. And some are known to regulate growth and other fungal interactions, like epiploe. Class 2 are systemic, so these ones are in the leaves, stems, and root. They are vertically transferred, which we'll talk more about later throughout the plant. And these have been shown, and it's believed that they help the plant deal with some stress tolerance. Next are foliar endophytes. These ones are locally restricted to leaves, hence the name foliar endophytes. These ones are I'm sorry, my slide's a little cut off. Horizontally transferred, I believe. But this particular group of endophytes is probably more well known for their medicinal compounds. In tropical areas in particular, there can be maybe hundreds of species of endophytic fungi in one particular leaf. And down there, when they have down in the Amazon, where there's much higher biodiversity, all of these fungi are competing with other fungi and bacteria and viruses. And so because they have this competition, they have evolved a big spread of different compounds, some of which we can make medicines from. And the last group or class of endophytes is dark septate endophytes. Dark because they're melanized, just like the pigment in our skin. Fungi will also produce that to help protect them from UV radiation and other things. Septate just means that every so often along the filament, there is a little separation so that there are different segments as opposed to one continuous little tube. Yeah, similar to mycorrhizae, in addition to them living on the roots, they can aid in nutrient uptake either directly or indirectly. Directly means the fungus gives the nutrients directly to the plant, and indirectly it can break down compounds around the plant that the plant could help absorb easily, because fungi are sloppy eaters. There's a lot of things that eat off of their sloppiness. Right, now getting more into my research. So my objective was to look at the roots of Pennsylvania sedge, which was a very common plant across the military base that I did monitoring work at, and then look at environmental factors that may influence a fungal community or total colonization. So the overview is culture the fungi from the roots and identify the most common species. Then I stain the roots and visually assess relative amounts of different fungi. This I did underneath the microscope, which we'll see later. The next thing I did was analyze soils for various things like nitrogen, phosphorus, water holding capacity, and then compare that to fungal data to see if there were any trends. And then the final step was to do DNA mass sequencing of the roots to figure out what other fungi were there that we did not pick up from culturing, such as mycorrhizae, which you don't culture on a normal petri dish. So first was identifying the most commonly cultured fungi from the roots. So I sampled 30 plants and cut up the roots and randomly split up the sections within each sample. And then I plated out 566 root segments and then monitored for growth. And as something would grow, I would cut out that, I would isolate that culture. And then as you can see from the picture, I had a huge tray of like hundreds of petri dishes. And I basically just looked for themes. I didn't know species at this particular time, so I used what I called morphotypes. So it's basically like, oh, it's pink and fuzzy. This is a particular morphotype. And then I grouped things that way. And then once I figured out which were the most common morphotypes, that's when I did visual identification as best I could, and then sent in pure cultures or DNA sequence pure cultures to have tested for confirmation. So the most commonly, here's a graph of the most commonly isolated endophytes. Fusarium oxysporum is not that surprising if you're familiar with these at all. Turns out Fusarium oxysporum is commonly isolated in a lot of cultures from endophytes. Part of the reason is it is easily cultured as opposed to some other things that don't culture as well. One of the species I would like to point out of interest is the second one called Acephala species CM7M4. This species had been previously sequenced in switchgrass from pine barrens in New Jersey. And I was sampling sandy oak barrens in Wisconsin and searching in the roots of another graminoid plant. So there's definitely some overlap in the habitat and the plant host that it was found. But other than this, it was not really previously described. So we kind of hit a wall. We were potentially going to publish this as a new species called Acephala herinae, which means sand loving because it's found in sandy soils. But yet genetic testing gets complicated, and we found that this falls within a complex. And so it was going to be a lot of molecular teasing out to argue that this is a completely new species. So we just kind of left it as is. Okay, the next step was measure the relative amounts of different types of fungi, which was dark septate endophytes versus arbuscular mycorrhizal fungi or ectomycorrhizal fungi. And this image is, as you can see, it's full of all different types of fungi, and it has very high fungal colonization from what you might expect. So here's a zoomed in. I know we've talked about dark septate endophytes a lot, but here is an image. You can see the dark hyphae, or this filament. And then if you look, I don't know, can you see, annotate? Yeah, thank you. There are these little darker lines, and those are the septa. So those are little separations that can help distinguish different chambers. And you can also compare that to the blue lines, which are thinner. Those are characteristic of arbuscular mycorrhizal fungi, and they don't have septa, and they're a little bit thinner, and they are not melanized. Next is staining and slide prep, or I guess is methods. So again, when I sampled my roots, I cut them into a bunch of roughly 1 centimeter segments, and then randomized, some of which went to culturing, some of which went to staining, and some of which went to mass sequencing. For the ones that I wanted to examine, I stained them with trypan blue and Sudan 4, which will highlight various fungal structures. And then I put 3 or 4 of the root sections onto a slide. And what I would do is I would line up the root underneath the microscope without looking in the microscope. And then once I knew that it was within the field of view, I would look in the microscope and see where the crosshairs were crossing. And it's kind of a method of like point line intersect monitoring. And so I'd randomly select these intersections, then I'd record, what is this crosshair going through? Is it going through, arbuscular, like hyaline hyphae, which is characteristic of arbuscular mycorrhizal fungi? Is it a dark septate endophyte? Or do I see clamp connections, which is indicative of a basidiomycota and an implied ectomycorrhizal fungus? My sample size was 1,800, so 1,800 times I looked into the microscope and took a sample or recorded the data. And like I said, overall results, big picture is there was surprisingly high fungal colonization, especially for a sedge, which was previously considered to be non-mycorrhizal. Here's a graph showing basically the same data: total fungi, septate, dark septate hyphae, hyaline hyphae, microsclerotia, which was a weird category that I found a few times that I had to add in later. And then the last category is clamps, which we'll see later. Clamp connections are part of maintaining a dikaryon that basidiomycota do exclusively. Another really interesting thing that we found is cohabitation of ectomycorrhizal fungi, dark septate endophytes, and arbuscular mycorrhizal fungi, all within a very small area. So this picture, the 3 arrows are pointing to the 3 types. The top arrow is pointing to ectomycorrhizal fungus, and as we'll see, we can point out the clamps. I think it's the next slide. We'll highlight it a little bit better. Yeah, so you can see here, this is a Tomentella species, we believe probably Ferrugenia, based off of the molecular data that we got. And Tomentella has very regular and characteristic clamps on it, which you can see on the image of the right. And the image to the left is the whole root tip, and you can see how this hyphae is kind of forming this net around the sheath. And I did my sampling fairly early in spring because that's when this, when Carex pennsylvanica produces its flower. And without its flower, it's really difficult to distinguish this from some similar looking sedges. So the fact that this fungus typically forms ectomycorrhizae with its host and hasn't fully formed a sheath on this root. We are not sure is a product of the timing of when we sampled, and maybe it's starting to fill in completely, or just the relationship of the plant and this particular fungus. It doesn't form the full sheath like we normally see in other plants. Yeah, so the plants totaled an average of 58% total fungal colonization, which is crazy high. So 50% of the time when I looked underneath the slide in the crosshair, there was some type of fungal structure in the way. Dark septate endophytes were definitely the most common type of fungus, which I expected going into my research. It was kind of a dark septate endophyte hunt, but I found a lot more. So they were found in 37% of intersections, but I think almost more interesting is how many had ectomycorrhizae unexpectedly. Phase 3 was soil analysis. So yeah, I did water holding capacity, organic matter, nitrogen, phosphorus, and pH, which all have different effects on soils and microbes and the reliance of a plant on a microbe. pH is a really good example. For phosphorus, phosphorus will bind to soils very strongly unless it's within kind of an ideal bell curve range of pH, which I can't remember exactly off the top of my head, but I know it's slightly acidic. So when soils are in that ideal pH range, phosphorus is a lot more soluble, and so plants don't rely as heavily on their mutualistic fungus. But if the pH of the soils are out of that range, it's harder for the plant to acquire those nutrients, which makes them more heavily reliant on their mutualist fungus. So not quite significant, but an interesting trend is total fungal colonization was positively correlated, or appears to be positively correlated with water holding capacity. Reasons for which would be skeptical, not exactly sure why. And then looking at dark septate endophytes versus arbuscular mycorrhizal fungi, it's interesting to look at the trends. The stars indicate that the data was significant. So comparing pH versus organic matter, so as the pH goes up from very acidic towards neutral, the total amount of arbuscular mycorrhizal colonization goes down. And you see the opposite trend when you look at dark septate endophytes. And similar when you compare organic matter, as organic matter increases, the total amount of arbuscular mycorrhizal colonization increases, whereas dark septate colonization is not affected at all by various types of organic matter. The last phase of my thesis research was doing DNA analysis using Illumina sequencing. And so basically what this does, if you're not familiar, is you're sequencing all of the fungal DNA that might be in that sample. And so in this 1 centimeter segment of root, there could be potentially dozens or more species of fungi in it. Unfortunately, with some of my amplification techniques and testing for how strong the signals are, we lost a bunch of samples. And so we only got data back for 18 of the 30 plants that we sent in. But it still provides insight. So here is bar graph showing the spread of the different types of bacteria, or sorry, not bacteria, different types of fungi. Ascomycota were definitely the most prevalent, which makes sense because most dark septate endophytes have Ascomycota affinities, and also some Ascomycota are ectomycorrhizal fungi. These Basidiomycota, I have to be careful with my wordage because some of these Basidiomycota could be pathogens or saprophytic fungi that were residing in the soil, but I did surface sterilization techniques on the roots, and so hopefully everything that was sequenced was actually growing inside the root. But I will just say broadly, Basidiomycota are basically representing the ectomycorrhizal fungi. Again, the glomeromycota represent the arbuscular mycorrhizal fungi. The zygomycota are probably dark septate endophytes. The Rosello mycota, I don't really know. That's kind of a weird fluke. I don't know much about them. And then a bunch of it was just unidentified because of not great sequencing. Okay, so putting it all together, it was interesting comparing the culture-based data and the molecular data to see what was common and different throughout both. It was nice to see that we had very high average colonization rates and some special species found in the roots. I was almost most surprised by not seeing huge trends in fungal colonization or community when we examined that or compared that to various environmental factors, because we believe that they influence them a lot more than maybe they do. Again, back to the culture-based and molecular data, Fusarium oxysporum was the #1 most cultured fungus, but it was very low and didn't show up in our most commonly sequenced fungi. And so I think it's good to do both so you can compare, because one way of sampling is a little bit more biased than others. I do think it's really interesting that the species that we were working on describing, Acephalorhyninae, was both, it was the second most commonly isolated OTU, which is basically like a molecular fingerprint of a species. And it was also the second most commonly isolated. So that is definitely associated with the roots of Carex pennsylvanica. Slide. Yeah, there we go. Compare environmental factors and other things that affect colonization and community. There were no soil differences between tree and no tree sites. One thing I failed to mention is, as I set up my study design, I sampled plants that were both growing directly underneath the canopy of a tree, which would have influence of the tree, and then just outside of the suspected root zone of the tree. There was no differences between those. We did see that there was correlation between pH and colonization of dark septate endophytes and arbuscular mycorrhizal fungi. When I did my plant sampling, I also identified all of the plant species in the surrounding area, basically one square. I actually used a hula hoop, so everything within the hula hoop I identified. And again, no correlations with either biodiversity or particular species and fungal biodiversity. And what we would expect is the more diverse the plot of plants, the more diverse the microbes, but we did not see that in our research. But we did see that the pseudo-ectomycorrhizae were more common in tree influenced regions, which is really interesting to think that trees and sedges are potentially sharing fungal species or sharing mycorrhizal species, either both directly connected like Chinese finger trap, or even just the same species in different, the same species interacting with such different types of plants in the same way. So Carex pennsylvanica associates with dark septate endophytes, arbuscular mycorrhizal fungi, and ectomycorrhizal fungi, which is interesting because it was previously thought to be just facultatively arbuscular mycorrhizal, which just means sometimes it does, sometimes it doesn't, depending on the situations. But yeah, it forms all of them, and sometimes in close proximity. And I think more broadly, it's interesting to show that all three of these types of fungi that reside in roots, or endorhizal fungi, do coexist with each other. And then just the general breakdown of abundance that I saw, the AMF were all Glomerulaceae, so it's a family within the Glomeromycota. Most of the dark septate endophytes were Halochiales, that's an order of Ascomycota, and the ectomycorrhizal fungi were mostly Tomentella, Russula, and Laccaria. Laccaria is, I think of it very associated with sandier soils, and so it's not surprising that we found Laccaria on these roots here in our sandy soils in central Wisconsin. So yeah, these are suggestive, showing that carex, which we previously thought to be non-mycorrhizal, is actually probably mycorrhizal. Mycorrhizal morphology is a product of both the host and the plant, or maybe the environment. Sand prairies, which is the particular habitat that I was sampling, they seem to be dark septate endophyte heavy. Maybe this is because they're highly disturbed, or I don't exactly know the reasons why. And as far as environmental conditions or soil conditions are concerned, the organic matter seems to be the most influential property affecting the fungi in the soil, or fungi on the roots. But I have to end with my scope of limitations. So I only sampled Carex pennsylvanica on Fort McCoy, which is a military base in central Wisconsin. I only sampled one plant species, so to project these findings onto other plants, we have to be skeptical. Most environmental factors, or another way to say is soil factors, were not significant. And so the true relationships between these fungal symbionts and the plant were not tested. A way that I could test it is I could have taken my fungal isolates and then grew them with the plant under various conditions and monitored that. If I would have gone for a PhD, I'm sure I would have had to do that, but this was good enough for a master's degree. All right, well, it's been years since I've talked about it, and it's fun to relive. Thank you guys so much for letting me talk about my project. And I think we are moving on to Kyle next.
Perfect. You can let her rip, Kyle.
Great. Well, Sam, thanks for talking about that. And honestly, Sam finished his project after I had already left. So this is my first time really seeing this this presentation. And we were talking before the webinar started, and Sam and I were just at the Telluride Mushroom Festival together, and we were talking about his research, and I really find it interesting. Even though he was looking more at like ecological systems, I think that this information has implications for agronomic systems as well, and just kind of highlights how little we actually know about plants and fungi and the interactions that are happening all around us. So, okay, cool. So I'm going to take what Sam said. I think Sam did a great job just of talking about endophytes and dark septate endophytes in particular. So endophytes is a bigger umbrella term. Dark septate endophytes are a particular type of endophytic fungus. So my title slide here is talking about endophytes and sustainable agriculture. And really, I'm going to be talking about fungal endophytes. So the term endophyte by itself can relate to a bacterium or a virus or protozoa or a fungus, all of which lives inside of the plant without causing any apparent harm to the host plant. So there are a number of implications of these endophytic fungi and how we as cultivators and regenerative agricultural enthusiasts, how we can maybe use some of these relationships to help benefit our soils and also our plants. So also, as Sam alluded to earlier, often as mycologists, we try to categorize various fungi into different groupings, and the term symbiosis often can make one think that it is a beneficial interaction that's going on, and really that's just one type of symbiosis. So a fungal mutualism is seen here on this graph in the upper right-hand corner, is going to be where both the plant and the fungus are receiving some sort of benefit from this relationship. The most common example of this is mycorrhizal fungi, where both the fungus and the plant are getting a benefit. The fungus gets benefits from the carbon that the plants are fixing and creating carbohydrates during photosynthesis. Well, a lot of those carbon compounds can be shuttled down to the fungus, and in return, the mycorrhizal fungus can help greatly expand the surface area of the root system and make the plant give the plant access to more nutrients that previously it was unable to access with just the naked plant root. Another type of fungal symbiosis, kind of the opposite, I suppose, would be a parasitism, where the fungus is getting a benefit by directly stealing nutrients from a living host, and the host is not getting a benefit, is getting harmed in some way. There are other examples. That there are certain plants that don't produce chlorophyll, that cannot photosynthesize, and they will actually steal nutrients from both fungi and the plant host that the fungus is infecting. And then another lifestyle that is common in the fungal kingdom is a saprophytic fungi, which are fungi that are going to break down dead organic material. So again, we kind of try to categorize the fungi, in these different groupings, but in reality, it isn't so black and white, and it can change over the course of a plant's life or over the course of a fungus's life. And I think it is good to at least be aware that these interactions are more on a spectrum and can change, and they're not so static. So that's kind of the major point that I want to make here. And also with endophytic fungi, oftentimes we don't really know what's going on. And I would say fungal endophytes, even though they were the term was first coined in the latter part of the 19th century, it's really been in the past decade or two that researchers have really started to look at endophytes more closely. And that's also due to the increase in analyses and techniques that science has available to us. So when we are talking about endophytic fungi, and in particular, so with my presentation, I'm really talking more so about crop plants and cash crops, or plants that people will be growing in some sort of agricultural system. So there are a number of direct and indirect benefits that these fungi, these endophytic fungi that live inside of the plant tissue, can ultimately give to the plant. So this one example of a direct benefit that endophytic fungi can give plants is that it can help, the fungus can help the plant in uptaking particular micro or macronutrients. So that is a big benefit that these fungi give to the plant. In addition, a lot of these endophytic fungi can produce either secondary metabolites or other compounds, certain ones being plant hormones or plant growth regulators that can ultimately influence how the plant is growing. I think there's still a lot we don't know. And I think that part of that is why, one of the reasons why Sam's research was so interesting is because we're kind of trying to unveil some of these relationships and interactions and ultimately determine what is going on. And then I think the next step is like, how can we apply this information to create healthier systems and healthier crops? Again, there are a number of potential benefits that endophytic fungi can give to plants. Some of the more indirect benefits that they can give is a production of what is called siderophores, and these can ultimately help chelate different nutrients and make them more plant available. Another big benefit that is commonly seen in the peer-reviewed research is that certain endophytic fungi can help particular plants endure against stressful times, whether that be high salt concentrations or droughts or extreme weather conditions, and also help protect plants against biotics, so other pathogens that may infect the plant. So that could be this fungal endophyte, or the endophytic community of a particular plant, can help protect the plant so it is less likely to get infected by fungal pathogens. Or another example, like Sam mentioned earlier, is these fungal endophytes can produce secondary metabolites that ultimately make the plant less desirable to herbivores or other larger animals that may be consuming the plant, and thereby decreasing the plant's fitness. So as this diagram shows, endophytes are really found in all parts of it's difficult for me to say all parts of all plants, but it's seemingly at least most parts of most plants, even plants that were previously thought of as non-mycorrhizal. So these fungi can be isolated from the leaves, they can be isolated from the stems, they can be isolated from the roots, or even the flower as well. So I think it's very interesting, and I think a lot more research needs to be done just on an ecological scale of what is going on with these various interactions, and what can we as growers or cultivators or farmers do to facilitate these interactions and use them to our benefit. So another part of endophytes that Sam talked about is the transmission of these endophytes. So how do the plants ultimately acquire these endophytes? And there are two major ways that we classify endophyte transfer, and those are going to be vertical transmission versus horizontal transmission of endophytes. So a vertical transmission of endophytes is going to be the parent plant giving or inoculating the offspring, the seed, with a particular endophytic community. Now, depending on the plant, certain plants require this endophytic fungus to be present in order for the seed to germinate. Sam gave an example of that. Another example is orchid seeds, which are very small and they don't have a lot of nutrients in them. Thereby, they need this fungal partner so that when the seed germinates, it is able to access various resources such as water and nutrients. So that's a vertical transmission of endophytes, whereas horizontal transmission is going to be more so once the plant has sprouted, it has biomass above the ground. Now endophytes can be transmitted via insect vectors, just from wind. They can also be transferred if there is some sort of damage in the leaf or the plant tissue. Then endophytic spores that are in the air can land and germinate and enter into the plant cell. So as far as agriculture is concerned, there are an increasing number of products that people use to coat seeds prior to germinating the seeds. And if this is done with intention and inoculating a particular seed with a known endophytic fungus, can really help give the plant or the seedling a little jump start, because it already has this endophytic fungus that is beneficial to the plant. It's already inoculated by the time that the seedling germinates. So we have this vertical transmission from parent to offspring versus horizontal, which are endophytes which are going to be acquired over the plant's lifetime. Let's see here. There we go. So especially in the past decade, there has been a lot of research done, peer-reviewed research about endophytic fungi and how they help various plant species. And I think there's a lot more work to be done. So I'm hesitant to make overgeneralizations, but they do seem to be found, endophytic fungi, that is, do seem to be found in all parts of most plants. They're definitely, they're probably not in all parts of all plants, but when we look for them, we commonly find them. As we progress as scientists and just as a scientific community, and our methods and analyses get better and more accurate and precise, we'll likely be able to understand these relationships a little bit better. One of the common ways, or especially historically, that these interactions or these fungi have been identified is through culture-based methods. And I think that can help, that can be beneficial, but it's also limiting at the same time, because not every single endophyte that is in a plant can be cultured. The perfect example is arbuscular mycorrhizal fungi, which cannot be cultured without its host plant. Another important aspect of this whole thing is that some of these endophytic fungi are more generalists and they can infect a number of different plants, versus some endophytic fungi are going to be more host specific, especially a relationship like the orchid seed, and it needs its mycorrhizal partner in order to germinate. That's going to be a very specialized fungus that is required for that relationship, versus a lot of these horizontally transferred endophytes that are just passed via wind or insect transmission. Those are typically going to be more generalists. So that's important also if we are looking to purchase a particular product that has an endophyte in it. It is good to know if this product contains an endophyte that is more of a specialist, where it's very important to know which endophyte you're using for your particular crop, versus some of these more generalist fungi. They could probably be used across a number of different crops. So just to kind of reiterate, there are a number of benefits that these endophytic fungi can give to plants and in particular agricultural systems. Some of those benefits include increased nutrient cycling. It can help increase the plant's defense against insect, mammal, and also fungal and bacterial pathogens. And this could be done either by the endophyte producing compounds or secondary metabolites that increases the plant's defense, or it could also be just this endophytic fungus being present in the plant biomass influences the plant to upregulate its own defense systems. So there are a variety of mechanisms for endophytic fungi increasing plant fitness and plant health and decrease plants' susceptibility to various pathogens. We also talked about these endophytic fungi producing different plant hormones or plant growth regulators, which can definitely influence the growth rate of a plant. Certain endophytic fungi, especially in the root system, can help increase remediation techniques. So there are two major types of remediation are phytoremediation, which is going to be using a plant to decrease pollution in a particular area, whether that be biological or chemical or otherwise. And then there's mycoremediation, which is using a fungus to break down a particular pollutant. And I think, those are kind of two distinct areas of bioremediation. But honestly, especially considering endophytes in this relationship, I think that they can both be used together in a phytomyco remediation technique where we are using plants that have known fungal endophytes that maybe help the plant absorb heavy metals and thereby can actually increase a plant's ability to remediate a particular area. Moving on, there are also a number of products currently available through agricultural supply stores that are actually endophytic fungi that act as biocontrols. So they can infect insects that would potentially be a pathogen against plants. So a common example of this, I think I go into in the next slide. Yeah, here is going to be Bavaria bassiana. And that is a entomopathogenic fungus. So a fungus that is able to infect certain insects. And we see here in the lower left, this insect has been infected with Bavaria. And now that insect is no longer alive. The fungus kind of consumes the inside of the insect and then is able to produce some sort of fruiting body to kind of spread its spores. The fungus is now going to spread its spores so that it can infect other insects. So it has also been found that this Bavaria species, in addition to acting as a biocontrol, it's a fungus that kills the particular insects. Well, this Bavaria fungus has also been found as an endophyte in certain plants. So that was previously unknown, and I think is pretty interesting. And yeah, I think there's a lot more research that needs to be done with about these different interactions so that we can better understand how we can use them to create more sustainable and regenerative systems. So yeah, as I alluded to, there are a number of products available that already contain endophytic fungi. They're just typically not marketed that way. Metarhizium is another fungus that can act as a biocontrol. Eiseria is, I think there was a taxonomic change with that. I don't think it's called Eiseria anymore, but it is another fungus that can act as a biocontrol agent. And I think as we learn more about these plant fungal interactions, I think that we can use them to create more sustainable systems and techniques and grow healthier crops that potentially have new molecules in them that can increase human health or benefit us in some sort of way other than, the direct nutrient composition of a particular crop. So collecting endophytes is rather easy to do. I mean, I guess it's all relative. But if you have a lab with a flow hood and you have all the equipment, it's relatively easy to collect endophytes by collecting a particular part of the plant, doing some sort of surface sterilization process, and then plating that plant material, which ideally the outside of this plant material is now sterile, and you'll put that on a particular medium in a petri dish, and fungal and bacterial communities will grow out from there. And then you can isolate these different fungi and then potentially grow them up and use them to re-inoculate your plants in the future. I think there are also other like lower technique methods that may be just as good as doing this isolation. This isolation may not be necessary, but it can be done. And hopefully after after my presentation, we can kind of get into and have a discussion about that and how we can use some of this information in regenerative agricultural systems. So collecting endophytes, that was something that Sam did by isolating them out and growing them out in culture. And yeah, there is a number of different research projects looking at plant-specific endophytes and how we can use these endophytic fungi to increase the health of particular plants. All right. So as we talked about, endophytes are this broad group. And then we are talking specifically about fungal endophytes, which is a little more specific. Sam talked about dark septate endophytes, which is even a more niche kind of area of these types of plant fungal interactions. And another one that I just wanted to mention briefly because it's new. I would say past 2-3 years. I'm not sure exactly when this was found, but this subphylum called Mycologist was previously unknown. And why I think it's so interesting is plants that previously were thought to be non-mycorrhizal, or they do not form these mycorrhizal associations. Well, they actually do, some of them do form mycorrhizal-like interactions with particular fungi. So these are called fine root endophytes. So these are kind of distinct from the dark septate endophytes that Sam was talking about. And one of the reasons I think this is so interesting is because previously we thought that a lot of these plants did not form mycorrhizal relationships. So in particular, this fine root endophytes, Mycologist, I think they were first discovered looking at liverworts and mosses, which are older plants, evolutionarily speaking. And yeah, so we are finding that originally we thought that they did not form mycorrhizal relationships, and now we are finding that they have more of a specialized type of relationship with endophytic fungi, and that is termed fine root endophyte. So like arbuscular mycorrhizae, these fungi are typically going to be clear, hyaline, and they do not produce septa typically, or if they do, they're few and far between. Okay. So that's how they're kind of similar, and a lot of the functions. So they help increase the surface area of the plant roots, and they're able to help the plant access more resources such as nutrients and water that previously the plants were unable to access. One of the major differences, which is key in my mind, between Glomeromycota, arbuscular mycorrhizae, and these fine root endophytes or Mucormycotina, is that a lot of these fine root endophytes we can actually culture without the host plant. So arbuscular mycorrhizal fungi, you need that host in order to grow out the fungus. Well, these fine root endophytes, you can grow in vitro in the lab without the plant host. So the more that we learn about these relationships, we could potentially create products or just on-farm methods of facilitating these relationships to improve crop and soil health and decrease the need for chemical fertilizers or pesticides and herbicides. So this is kind of a newer grouping of fungi, these fine root endophytes. So not a whole lot's known about them, but I just wanted to talk about them and hopefully create awareness around these new things, because the field of mycology is not, I mean, fungi have been studied for a long time, but typically they were studied under the plant pathology or in the botany departments of particular schools. And yeah, now I feel that mycology and mushrooms in particular are kind of trendy. They're in mainstream media often. And yeah, I think that mushrooms are a great gateway to make people more interested in mycology and fungi in general. So this is just a new type of plant-fungal relationship that previously was unknown. Another example of something that was not taught to me when I went to school to study plants a decade ago is something called the rhizophagy cycle. And this was first published by Dr. James White at Rutgers University in New Jersey. And briefly, I will go over it because I think it's interesting. It doesn't particularly pertain to endophytic fungi, but it just does go to show that ultimately we do not know everything, and there's a lot of things that we do not know. For instance, this rhizophagy cycle where so the plant is going to be releasing exudates out of the root tip, and these are often carbohydrates that the plant made in photosynthesis that then it's shuttling out of its root to attract particular microbial communities. So the rhizophagy cycle is actually where the plant root kind of quote unquote eats or consumes the bacterial communities or individuals that are near the rhizosphere. So the bacterium actually enters into the plant cell, and then the cell wall of a particular bacterium, if it has a cell wall, will degrade, and the plant actually will siphon off some nutrients from this bacterium. And then afterwards, the plant root kind of spits out this bacterium that still has a plasma membrane, and then it goes back into the soil system where it will continue to grow and reproduce and get nutrients, and then it will get cycled back into the plant root. So that's what the rhizophagy cycle is referring to. That's kind of just like the shorthand abridged version of it. But really the point I'm trying to make here is that there's a lot that we don't understand about plants, and I think that research like Sam's goes to teach us so much more about these interactions that are happening all the time, and then ultimately how we can use this in sustainable and regenerative agriculture. So as far as future research goes, I think we need to, there's a lot of just basic research that still needs to be done about the roles, the various roles that fungi can play in the soil food web. We didn't even talk about soil or fungi in the soil, really. Endophytes, we're talking about fungi that are in the plants. And then a whole nother thing is the soil food web and what fungi are doing in the soil and how they interact with other organisms in the soil system. I think future research with endophytes and plants could also help create various products that are crop specific, with particular endophytes that are known to infect only certain crops, or even just regional microbial products that say are good for the Pacific Northwest versus a product in Colorado that contains very different endophytes because those are already adapted to that bioregion. So as I have alluded to a number of times already, just understanding these relationships between plants and fungi and realizing that it can be more on a continuum or on a spectrum where it's not always, oh, Fusarium oxysporum is always a plant pathogen that's going to cause the vascular wilt. Well, we also find that as an endophyte in plants that are not displaying symptoms of a Fusarium wilt type pathogen. So what's going on there? I don't think that we have solid answers in science. So something I'm also interested in is we were talking about plant fungal endophytes. Well, with personal communication between me and Dr. James White, who is talking about the rhizophagy cycle, and something that I have experienced just hands-on working with plants and fungi, especially with fungi, is, okay, so plants have a community of microorganisms inside of them. And fungi, the fungal hyphae, filamentous fungi, also have a community inside of them. And oftentimes this can be bacterial, or there can be like a bacterial community around the filament as well, whether they are siphoning nutrients off of the fungal hypha, or maybe they're there to help protect the fungal filament. And even smaller, I think that viruses can be in both plants, but also in fungal mycelium. And it just kind of gets almost like a fractal, where we just see these smaller and smaller communities inside of plants, inside of fungi, inside of us. Obviously, we have the human microbiome. And yeah, so I think that there is a lot more research that can be done with that, in particular, as our techniques get better. And another thing that is talked a lot about in the regenerative agricultural scene is disease suppressive soils and creating a soil environment that makes plants more resistant to particular pathogens. So what role do fungi play in disease suppressive soils, and do endophytic fungi have a role in that as well? All of these, I think, are more just like hypothetical questions and areas for future research. So yeah, I wanted to keep it kind of short and talk about more of the applications of how we can use these endophytic fungi in regenerative systems. And honestly, we probably already are, whether we know it or not, because these endophytic fungi are everywhere. In particular, if you're growing from seed, oftentimes these endophytes can be transferred horizontally or vertically from the parent to the offspring via seed. So as soon as the seed germinates, the plant is infected with endophytes. The various endophytic fungi could potentially be very important and key to crop and plant health and resistance to particular diseases. There are a number of products that already contain endophytes in them. I don't want to say any brand names because I'm not sure the efficacy of particular products, but Bavaria and Metarhizium and Eiseria are all examples of fungi that are found in products that we have since discovered are actually endophytic and can live part of their life cycle inside of the plant. Something we were talking about before the webinar, and I hope that we can get into a discussion about this afterwards, is potentially inoculating plants just with compost teas or compost extracts. Perhaps this is a low-tech way of inoculating plants with endophytic fungi to give them more of a head start against potential pathogens. Also summarizing, I want to create awareness around using fungicides. Most fungicides, if not all, are broad spectrum, and they do not just kill fungal pathogens. They kill all fungi, and that includes endophytic fungi that could be giving plants some sort of benefit. Yeah, I have a number of other examples I can go into. I work typically with cannabis plant. That's cannabis indica, cannabis sativa, hemp, or recreational marijuana, whatever you want to call it. And with cannabis, botrytis cinerea is typically, there is a commonly seen pathogen. Well, recently it was also found that this same fungus of botrytis can actually be an endophyte of a number of different cannabis plants. So maybe how the plant is interacting with this particular fungus changes over over the lifespan of a particular plant or of a particular fungus. So the point I'm trying to make here at the end is that using broad spectrum fungicides that may damage your fungal community in your plants and in your soil. So just be careful and be intentional if you need to use those in your farm. So with that, I would like to thank the Soil Food Web, Dr. Elaine Ingham. I think she has been a great advocate for soil biology and just creating more awareness around these different organisms, and also using a microscope to try to help quantify some of these organisms. And I think really just looking under the microscope at soils, once you have a microscope and you understand how to use it, you can look at whatever you want. And I think that gives us a broader understanding of the world around us, because oftentimes we don't see this stuff with our naked eye that we can see under the microscope. So it just can help create more awareness. So I'm very thankful for the Soil Food Web School. That's the point I'm trying to make here. And Sammy in particular and Adam, we met at the Radical Mycology Convergence I think it was last year. And yeah, I'm very thankful for them for hosting us, for having these webinars, and just allowing us to talk about some of these less known aspects of plants and fungi and how they interact. A big thank you to Dr. Tom Volk and Todd Osmondson, who were really the ones who I came into it very interested in mushrooms and mushroom physiology and how those grow. And I think that both of those individuals helped teach me that the field of mycology is much more vast than just mushrooms. So yes, I'm eternally grateful for those two individuals. They've been very influential on my life and my professional career there afterwards. If you have any questions, you can email me directly at kyle@mycophyte.com. I do currently have a biological consulting company here in Eugene, Oregon called Mycophyte Solutions. You can find us at mycophyte.com. And with that, I would like to end my presentation and hopefully now have a discussion between Sam, Adam, Dr. Ingham, and I just about endophytic fungi and some of the implications that they have, whether it be in the soil food web or making compost, or just how we can maybe use this information to further benefit our agricultural systems.
Awesome. Thank you so much, Kyle. And we do have, I'm going to reshare screen here so that I can update, because we will have some questions that have come in from the audience. So let me just switch over to that. While I'm getting that queued up, did you have any thoughts that you'd like to add here at the top, Elaine? I always want to give you the chance to express what you're thinking.
There's probably 3,000 different questions and answers that to get involved in any one would be take the rest of the day. But just that getting the proper sets of microorganisms back into your compost, which for us is the way to get those organisms back into the system. We want to make certain that when we are composting, we are selecting conditions that will prevent any of the disease or pest organisms from being able to grow in that compost. So there's a definite reason why we require things to be done in a certain way, because in 24 hours, you can make perfectly normal, good compost. You don't have to be waiting 2 or 3 months waiting for it to mature, and the temperatures just never come down. So getting all those organisms growing in that compost, you have to have a huge diversity of food. You can't just grow microorganisms on 1 or 2 feedstocks. You've got to pick something that has, pick a set of starting materials to get things going, and that's just an absolute. Then adding worm, worm compost, you get an increase in species diversity of the bacteria, the fungi, the protozoa, the nematodes, the microarthropods, things that should be in that compost as long as you have chosen the proper conditions to kill the problem organisms. And so after that, you can let your microbes grow to your heart's content because they will be the beneficial organisms. And see, I can go on for the next 3 weeks talking about this. So I'm going to turn it back over to you, Adam. I'm sure there's lots of other questions.
No, I think you've actually, Elaine, set us up really well for this discussion and this Q&A session with comments about compost, because compost is one of the big tools, and not just any compost, right? Biocomplete composts that we teach here in the foundation courses and our advanced programs, so that people can hit those, I like to call them sweet spots, or the Goldilocks zone, right? Keep that compost as it's cooking in really optimal conditions, so
Not
that it
too
becomes
hot, not too cold. Does it sound like Goldilocks? Not too big, not too small. Yep.
Yeah, so that's perfect. All right, what I think we'll do here is, Sam, we haven't heard from you for a while, so I may start with you and then Kyle and then Elaine for comments on these various questions. But because of the timing, I just want to let people in the audience know, I think we'll extend this just another 15 or 17 minutes. Elaine, I know you have somewhere else to be as well, so we'll let you out in time or whenever you need to go, of course. I'm not going to tell you what to do.
Live dangerously, boy.
So the first question we got was, could endophytic fungi be effectively applied as a foliar spray or soil drench to improve the growth of crops in regenerative agriculture? What do you think about that, Sam and Kyle?
Yeah, I'll jump in. I think for sure, I mentioned some of the research by somebody else in our lab, John Carver, and he was looking more at this. I think it's tricky because, yeah, complicated answer. I think it'd be tricky to do it in a very precise way because, as we've seen, the diversity of endophytic fungi is huge. So you would have to sample, all 100 or 200 species of endophytic fungi and then do tests for all these different things with whatever plant host you would want to figure out the exact benefits. Or you could do it in a more simplistic, low-tech way without fully understanding what's going on. And I've talked to some organic farmers who actually have been doing this, and yeah, they didn't understand the mechanisms of why, but basically when they were growing a particular crop, they would take some of the ones that did really well and they would blend them up in a water mixture and then apply them to the next generation of crops in hopes that something in that plant was going to transfer over. And so it might be one species, it might be a collection of species. We don't really know. So I think that it's really easy to do low-tech endophytic inoculations, but there would need to be a ton of research to figure out, say, like this species alone or this group of species will help with this. Sorry, that's kind of the best answer, but
that's excellent. And if I could jump in there just for the audience that is familiar with a little bit more of like our methodology here, this is why we really focus on getting diverse local populations of these organisms back into our system. A lot of times the diversity and abundance of the native microorganisms have suffered because of how we've managed the soil in the past, especially if people have utilized a lot of agri-inputs or tillage or other things. And so we're trying to, with the compost piles, grow out the organisms that are very beneficial to the plants that we want to produce and get those populations up and then inoculate that at planting and, sometimes at other times as well. But I think it's a similar thing though to what you're saying, Sam, is we're doing a diversity protects us from pathogens kind of mentality here. Instead of knowing exactly what endophytic fungi we're putting in, we're trying to get a lot of different fungi into that system. So something will be able to compete, consume, inhibit the organisms that could cause harm to our crops.
And I think part of the reasoning there is we've got to get people converted over. We've got to get them to stop buying the things that kills these beneficial organisms in the soil. You lose your ability to build structure. You can't hold water. You can't suppress weeds. You can't suppress the diseases and the problem organisms. You don't get soluble nutrients from your own soil if you don't have the right sets of bacteria, fungi, protozoa, and nematodes. So it's really critical that we get going on this project now, even though we may not know the exact name of each microorganism that is being active this morning for these 20 minutes, but it's going to be somebody different after 20 minutes. And then in another 10 minutes after that, it's a different bacterium or a fungus or a protozoan that's going to reach the height of their population. So the complexity is just, we've got to do our best at controlling conditions to make sure things will remain right, and then be doing the research work that finally helps us figure out how we get these organisms into the system at the proper points that they will grow on this medium and reach this level so that it can eat this other set of microorganisms so that the plant is getting the nutrients that it needs from those interactions. We have a little bit of work to do.
Brilliant. No, that says it very well. It's like the environment, the organism, the plant, these all are interacting all the time. And so natural systems are just too complex for my brain. Let me tell you what. Kyle, did you have any thoughts to add on this one?
Yeah. I think, like you had said, I think an overarching theme in biology in general is just that diversity is key and that it helps kind of create resilience, because if there is a particular instance or environmental event that happens, maybe by having a diverse group of organisms there, then hopefully, it's kind of like the shotgun approach where hopefully something will persist even if other things die. Mm-hmm. To answer the question, I would just say yes. It is my answer. But really, to the question listed here, but yeah, I think there's a lot more that we need to know. And I keep going back to what Sam was saying about how he cultured Fusarium oxysporum as an endophyte. Mm-hmm. I mean, we kind of have to rethink then how we word these things. It's like, well, that's also like a super common pathogen that can cause devastation among crops. So is it a different strain of this particular fungus that causes disease? Is it maybe there's an absence of a particular microbial community in the plant that makes it more conducive for this fungus, Fusarium in this case, to attack the plant and steal nutrients from the plant instead of growing in a symbiotic, mutually beneficial way? Yeah, I'll stop there.
I want to jump in on what Kyle said specifically with Fusarium oxysporum. I know that is one of the fungal species that is under what's called the complex. So there are probably a dozen or so species that when we do molecular sequencing, they all pretty much come out as Fusarium oxysporum. But with what we have for technology and analysis, it might actually be a dozen species of Fusarium oxysporum, which span a range of virulent to not. It also might be harmful in some plants and not others. So Carex pennsylvanica, maybe Fusarium oxysporum doesn't do anything to Carex pennsylvanica, but it may do something to other things. And so just having something that can hurt other things present in you can benefit you because it's kind of like a defense in a way.
No, that's a wonderful point, Sam. And it's like, I've said this in many webinars because I see that there's sometimes a pressure of like we want everybody to show up for the webinar. I'm not saying anybody's doing it wrong, but there's sometimes feels like a pressure like give us an answer. And I'm like, we have to be careful. Sometimes there are answers, and sometimes the only answer is you're going to have to experiment, right? Because I can't predict the conditions that you're growing in. I can't predict in your crop and other things. And so that kind of practical on-farm trial is what I want to see people doing more and more to become their own investigator of whether or not, you know, this method will enhance what's going on their farm. And then there are other things like diversity that I'm like, yeah, that's basically a universal answer. We want to see a lot of diversity in your soil food web.
I agree. And I think the best way to answer this question would be like, well, apply an endophytic fungus or a community of endophytic fungi to your plants and see if you notice a crop response. But as an agronomist, it's difficult to convince farmers to do that and to like keep these things separate and then measure the yields and the real impact that these applications are having. So yeah, it gets messy.
Sure. Let's make sure we get a few more questions in here. V says, are there examples of endophytic fungi inoculation being used for post-fire myco or phytoremediation projects? I think because Maui is in the news and other things, people are wondering about the potential here.
I can't remember the name of it, but I remember, I believe it was Tom Brunn's lab. They were looking at fungi that are prevalent post-fire. I'm not exactly sure what exactly they do in terms of remediation, but it was interesting to look and see, depending on fire temperature and severity, a lot of things die off, and seeing like maybe the couple things that survive. And I think what fungi do post-fire is mostly like they start breaking stuff down, and they're basically trying to reset the system, especially stuff that was severely burned. So I couldn't tell you an exact one, but I know that fungi do play a role in post-fire succession.
We have been applying compost extract from the Lahaina area of Hawaii after the fire, and they've done a really good job of selecting a wide diversity and applying it to that 100-year-old banyan tree. And the banyan tree is starting to reflower. Not normal after you've been burned to the ground, basically, but there was enough protected area on the tree that I think we're gonna rescue that tree. And you know, there's the control is here's the area where we didn't apply, here's the area that we did, and look at the difference. We've got 24 little shoots starting to come off, and nowhere else is that happening. So we need examples like that. We need to go out and have these the biology put into the forests or the shrublands or the grasslands, wherever, and have an area treated with the proper biology and an area that's not. We saw this back in the Paradise fires in California. The only people who didn't lose their homes were people who had put on compost and compost tea on their property. Their neighbors burned. They didn't.
Elaine, I'm curious, with the compost you put on the burnt banyan tree, because Hawaii is so isolated and has so many endemics, for people in Hawaii, do you always recommend using localized compost? So you didn't import compost from the US, you just make it on site so you have more.
Just too dangerous. It's just too blessed dangerous for us to do that, because what if we did bring a disease-causing organism in that could not be stopped by any of the microflora present in Hawaii? Our names would be mud for the rest of forever, and rightly so. So yes, we have friends who make compost there, and it's starting to enhance the opinion of the general farmer about compost. You've got to do it correctly. It's got to be done right. You have to be selecting for the beneficials and against the disease causers.
I actually saw a news report about the banyan tree resprouting last night, Elaine, and I kept waiting for them to talk about compost, and they never did. So that was a little disappointing.
Yeah, never did, even though that's the answer to get this banyan tree to regrow. But yeah, it's aggravating. But hopefully there was a series of gosh, it's got to be about 2 weeks ago, maybe longer. How time flies when you're having fun. That it was put out in the airwaves that that's how they were getting the greening up of the tree was to get the right microbiology in there. So we can only stand around and watch while Mike is on Hawaii right now, working with them and making sure that they're growing enough good compost to be able to continue to apply to the banyan tree.
That's awesome. Yeah, that was actually one of my first skepticism tests for the soil food web school when I got here, Sam, is I was like, all fungi of a certain species are not the same. Local populations of fungi get local adaptations to their environment. And so I was like, what are we doing? What are we telling people to do here with that? And then Elaine started expressing this like bioregional worldview, which I totally agree with, that, within a particular area, and that can be very geographically small in the case of some islands, and it can be relatively large in the case of some land masses, but you look at those things that created isolation, you say those local populations are going to be the ones we want to get back, not necessarily and probably not some commercial product off a shelf. I have not yet come across the commercial product I could endorse. Let's put it that way.
No, you can't, because what did you do to the diversity when you put it in a jug? You've got to seal it closed. It's got to be able to stay on the shelf for years if necessary in order to bring profit. So no, bugs in a jug are not an answer. You've got to keep your own compost going, and so either find somebody who makes really good compost, check it, test it, get your own microscope and be able to assess whether the organisms are present that your plants need or not, and now you know what to buy and what not to buy.
Perfect. I think we can get one more question in here. I want to be respectful of people's time since we are a little bit over, but I thought this was a really interesting question that we could talk about together. With agrochemical corporations starting to use biologicals, products like Elaine is talking about, off-the-shelf things, what are concerns and observations that we should all be aware of? I see promise for good practices, but also potential reasons for concern from genetic manipulation.
Yeah, and I think we just have to have regulations where they can't be just isolating some organisms from the area around themselves and just counting the number of organisms, diluting it to the right place, and then putting a whole bunch of data on the shelf. When we look at products that are produced that way, within just a couple weeks, you will go in and look at what's the biology in that bugs in a jug, and you can't find any. They're gone. I think there was a really good protozoan that was put into the jug with them, and that protozoan ate all the bacteria, and what do you have left? Even the protozoan's dead because it didn't have any more food. So you can't, 50% of those bottles that are on the shelf don't have any decent life in them. Maybe spores of a whole bunch of things you don't want to know about, but it's definitely not what's on the label.
how in the industries that you've worked in and doing crop consulting, do you find that there's a lot of pressure to get these biological products that are arriving in this marketplace?
Yeah, I don't watch the market that closely. I'm more, the research scientist and out in the field. Um, so I think I'm going to have to give that back to somebody else who might have a better idea.
Sure. Sam, Kyle, do either of you have you had experience with working with producers that are keen to use these biological commercial products?
Kyle?
I was kind of waiting because I have a lot to say about like all these things. So it's like, well, okay. So I think a lot of farmers that I work with in particular want an easy answer. They want to be able to go to the store and buy a product so they can apply it. And it's like, well, you should really be using biocomplete compost. And it's like, okay, where do I get that? And then it's like, you make it. But people don't want to do that. They're like, I'm already so busy growing my crops, doing my thing, that I don't want to add this whole other aspect to my system. When I think ultimately it can like save them time and money, but that's not the answer that people want to hear typically.
You've got to get them through year one, and they've got to see that they're actually doing less work, and they're not running the tractors, they're not running all of the different things that they have to. So we've got to get over that hurdle of first yearism, where we need to give them they need to give us small areas on their property where we do the proper management. And then all they have to really come back to us for come the next spring is an adequate amount of compost. And if we're putting out 1 ton of compost per acre that they're applying, and that will be in the second year, that'll be 500 pounds, and then the next year it'll be 250, and the next one, why bother? Because you've got the biology in your soil. So we gotta teach people about this.
I'm gonna change this a little bit and make it a specific example and kind of direct it more at Adam, because this is kind of more of your expertise. Working as a horticulturalist, one of the soils that we get it's advertised to contain mycorrhizae in it, but it's all just one species, like glomus, I'm pretty sure. And I know that it's kind of a generalist with a lot of things, but do you want to talk more about that and using this one particular mutualistic fungus in basically everything that you plant?
Yeah, I'm really concerned by the mycorrhizal products, and I actually have a paper where we assessed several commercial products and found that in some cases they facilitated invasive species more than native species. In some cases, they caused production reduction in the species that we put them with. Now, in that context, there was also the local population of mycorrhizal fungi, so we're maybe creating like competition. But I like to tell people, to me, getting a commercial product of any kind, including mycorrhizae, if you threw like 5 chihuahuas out with a pack of wolves, would you expect the chihuahuas to survive and do well with that pack of wolves? Like strains that are laboratory based, they're sometimes the ones selected are selected because they're easier to grow, not because they confer more plant benefits, and their genetic diversity might be very low, as you mentioned. And I just have my doubts about whether or not in a field scenario those kinds of things will persist. The other scary possibility is that we put something in that's kind of like below ground kudzu, and it does really well in that environment. Like you said, introduce something novel, and it knocks out local populations of those organisms. And, I know mycorrhizae is globally distributed, and many of things like glomus exist almost everywhere. But that's that local adaptation again, that when we put together plants that have been developed within that local context, soils that have those local conditions, and microbes that have that local population history, we tend to see the most synergy in those kinds of pairings. That, like, sometimes we call it home, home, home versus home, away, home, or home, away, away, or away, 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 right? So these experiments have been done, and there's a striking influence from putting together microorganisms, soils, and plants that are all from that local area, that bioregion, just naturally being more resilient and productive. I think Elaine had to go, by the way. So did I answer your question, Sam?
Yeah, for sure.
Okay.
I would like to add one comment to something you said, which I found really interesting. You said using this one commercial bought inoculum is actually more beneficial to invasives. One of the properties of invasive plants is their ability to associate with a wider spectrum of fungi, which is why we don't really see orchids that are invasive, because they have a very specific type of obligate fungus.
That makes sense. And in fact, in the tallgrass prairie, I did some work with an invasive Eurasian grass that seemed to reduce the diversity of the native mycorrhizal fungi after it had come into an area. And we weren't completely sure the mechanism, although we thought it might be a chemical, and it might actually be knocking back or just not supporting the things that are really important to our native species of tallgrass prairie plants. So even when you clear out the invasive grass, you can have like a legacy effect in that soil biological community that you need to correct. Yeah. Kyle, did you get cut off there? Because I want to let you complete your thought, and then I think we'll probably have to wrap up here.
No, I mean, I could keep talking about this stuff for the rest of the day, but I don't think there was anything pertinent that I really needed to speak on, and yeah. But I have enjoyed this conversation. I enjoyed just listening to Sam's presentation. I've heard about it, but I never saw the actual presentation. So it's nice just to get a little more background information on that, and also increase awareness around this, because oftentimes papers that are published, it's like they're not very accessible often. Well, whereas both Sam and my thesis and other people in our labs are available. Feel free to email either of us if you are more interested in reading the study. But really, I just wanted to increase fungal awareness around regenerative agriculture. So Adam, Sammy, Dr. Elaine, thank you all for having us on and doing this webinar.
Awesome. And you both have gotten a lot of love in chat for your presentations. People really felt that it expanded their awareness. It expanded my awareness of some things. There's a couple questions we didn't get to that I thought, man, I hope people will send those to the info@soilfoodweb.com address as questions so that I can answer them later, or maybe reach out to the two of you to get answers. But yeah, it's wonderful. And thank you so much, Sam, for joining us for the first time.
Of course, it was fun. Thanks for having me.
Awesome. Well, I think with that, we should sign out because we are over time. And I know everybody loves the Q&A. I would have loved to have gotten through about 20 questions today, but that's the haps. So thanks everybody for joining us, and to our presenters, to our support staff who have made this possible from a technical angle, organizing questions, organizing the Zoom, and all of the background stuff that goes on. We thank you very much. And with that, we'll sign out. And see you all next time.
Don't forget to click that like button,
Cheers.
Subscribe to our channel, and ring the notification bell to stay updated with all our new videos.